Good evening. Today is the last class for the NPTEL course Pharmacognosy and Metabolic Engineering. Today we will recap a few questions from all the 12 weeks that would cover the essential topics that were discussed in this course. So first the question for week 1. Which among the following methods are used for separation of volatile metabolites? Gas chromatography, ion exchange chromatography, affinity chromatography, and HPLC. Okay, so in this course we were dealing with plant metabolic compounds, and these plant metabolic compounds were majorly volatile. So because they were volatile, it means that their solubility in a given solution is less. Among these chromatographic techniques, ion exchange chromatography is used when you have a molecule that exhibits different polarities at different pH, which means it should be soluble in a solvent. Affinity chromatography could be used in the cases where you have a specific chemical interaction between two different molecules. For example, nickel NTA and histag. HPLC is used when the compound is soluble uh, in a given solvent, and then it can bind to a matrix. Because you're talking about volatile metabolites, the correct answer for this question would be gas chromatography. So here is a picture of how this chromatography would work, where despite using it in the gas chromatographic chamber, you also have this matrix to which our compound of interest would bind. So for this we have a carrier gas, and we would inject our compound of interest along with the carrier gas. So the carrier gas is what would allow for movement of these volatile metabolites, and within which we have an oven. So within the column it will get the compound will be retented and will have different retention timings. These different retention timings can be detected, and the signal can be amplified and viewed as chromatogram. Shown here is ion exchange and ion exchange chromatography, where depending on the pH you will have different binding affinities. The next question is, identify the correct pair of chemically transformed isoquinoline alkaloids among the following: reticulin and emetin, emetin and oxycodone, scopolamine butyl bromide and oxycodone, buprenorphine, buprenorphine and oxycodone. You can take a couple of minutes to think about this question, and then we'll come back to this question. Okay, so we are talking about isoquinoline alkaloids. These are a class of alkaloids that are derived from benzyl isoquinoline precursors. So here we'll discuss each of the options. First is reticulin and emetin. So reticulin is a benzyl isoquinoline alkaloid, but emetin is a protoalkaloid. It is not directly chemically transformed isoquinoline derivative. So this pair can be rejected. Second question is emetin and oxycodone. Again, earlier we saw that emetin is a protoalkaloid, whereas oxycodone, it's a semi-synthetic opioid that is derived from thebaine. And this thebaine is a benzyl isoquinone opioid. So again, here one of the options is correct. So oxycodone is a part of the right answer. Next, we'll have to see option 3. Scopolamine butyl bromide and oxycodone. So scopolamine is a tropane alkaloid. It is not a isoquinoline alkaloid, which means that this pair is also incorrect, although oxycodone is the right one of the right options. Finally, buprenorphine, it is again a derivative of thebaine. Earlier we saw oxycodone, which is also a derivative of thebaine. So both of these derivatives of thebaine form isoquinoline alkaloids. Right. The next question is, which alkaloids were most abundantly obtained in the top 1 mutant of poppies? Option A, codenone and morphinone. Option B, thebaine and odipavin. Option C, codeine and morphine. Option D, morphine, heroin. Okay. So here, top 1 is involved in these two processes, which converts thebaine to neopenone and oripavin to morphine. So in this Nature article, what they observe is when you have these top 1 mutant compared to the control, we have a lot of thebaine, but we have a minimal of oriparin, but no morphine and no codeine. So codeine we get from neopen, right? So only thebaine is present and oriparin is present. So when when there is a top 1 mutant, only the precursors are available, which is thebaine and oriparin. The next question is, select the terpenoid indole alkaloid that is synthesized inside the transgenic yeast upon expression of streptozidin glucosidase from Catharanthus roseus. Option A, cathinamide. Option B, tabersonin. Option C, vinblastin. Option D, catharanthin. Okay, so here we are talking about expression of a glucosidase. So glucosidase will break the bond that is covalently linking glucose to any moiety. So now we are talking about this gene from Catharanthus roseus, which is a plant that is expressed in transgenic yeast cells, and it's involved in terpenoid indole alkaloids. Okay. If we see here, we have streptozidin. Now, streptozidin has a glucose moiety, and this SGD is the enzyme called streptozidin glucosidase that removes this covalent linkage. Now, going back to the question, they are asking select terpenoid indole alkaloid that is synthesized upon expression of streptozidin glucosidase. The options are cathinamide, tabersonin, vinblastin, and catharanthin. We have tabersonin. That's the answer. Tabersonin. Okay. Here I have again given a detailed explanation for what streptozidin is, what streptozidin glucosidase does, how it leads to tabersonin. Okay. Next question is, strong activation of CR-GATA1 takes place in the presence of light. Based on this statement, identify the false option from the following. Strong activation of CR-GATA1 suppresses the light-sensitive genes. CRPAF1, the transcription factor, degrades in the presence of light. Strong activation of CR-GATA1 enhances windowlin formation. The presence of CRPIF1 influences the activation of CR-GATA1. So here we are talking about activation of a gene in the presence of light. So it's a light sensitive gene. Now we are also talking about false options among these four options. First, let's look at what is the function of this gene and how this light sensitivity plays a role. So if we see here, there is the CR gata 1 that gets activated in the presence of light and activates other genes like D4H, D8D, that is the one that's involved in D-acetyl, windolin, and so on. Now, this CR gata 1, it can be inhibited by CRPAF1. So, imagine it this way. In the presence of light, CR gata 1 will be activated, but CRPAF1 will be inactivated. Only when CRPAF1 will be active, it can inactivate CR gata 1. So both cannot function at the same time. Okay. And the enzymes that CR gata 1 can activate are D4H, DAT, the ones that produce windolin. Okay. So coming back to the options, strong activation of CR gata suppresses the light sensitive genes. No, it actually activates the light sensitive genes. CRPAF1, a transcription factor, degrades in the presence of light. True. We observe that CRPAF1 is inactivated in the presence of light. Strong activation of CR gata 1 enhances windolin formation. So when CR gata 1 is activated, we have D4H, DAT and all these enzymes that was needed for production of windolin. So that is also true. Finally, the presence of CRPAF1 influences the activation of CR gata 1. Yes. In the presence of light, CRPAF1 will not be active. It will be degraded and inactive. But if CRPAF1 is present, then it will inactivate CR gata 1. Okay. So the right answer for this question is strong activation of CR gata 1 suppresses the light sensitive genes. And again, this has been given in detail, explained here. The next question is, CR-MYC1 is expressed in co-transformed hairy root cultures of Catharanthus roseus. What was the effect of this transformation on the levels of terpenoid indolactylate biosynthesis? So, and four options are given here. Ajmalsin and catharanthin content increased equally. Ajmalsin and catharanthin content decreased. Catharanthin content increased more than ajmalsin. Ajmalsin content increased more than catharanthin. So you can think about this question for a couple of minutes. This is directly from a paper. We'll discuss it later. Okay, so we look at where exactly it plays a role in the pathway and the data from the paper. So CR-MYC1 overexpression, here they took two cell lines and tested the effect on both. One is the MY3 cell line, the other is the MY1 line. Both have overexpressing CR-MYC1 and they have a control as well. So if we observe here, compared to the control, in the MY3 line, there is multiple fold change, but in MY1 there is relatively lesser when you have CR-MYC1. As another experimental control, the SGD gene, which is the strictosylin glucosidase, that was also used. Now, why do they use two different lines? One reason is because they wanted to compare and see how in different lines, different transgenic plant cell lines, if there are differences in the levels of the metabolite according to differences in the cell lines. They wanted to see if it was universal or if it would vary with cell lines. That's the reason they used this. So they observed that in MY3 line it expressed CR-MYC1 expressed more. Then they measured the levels of ajmalicin and catharan. So the question here is now whether ajmalicin and catharanthine increased equally, or if one of it decreased and one of it increased. So what we observe is that in both the cell lines MY3 and MY1, compared to the control, we have more of ajmalicin. But if we look at catharanthine, in one of the cell lines it was more, in one of the cell lines it was slightly lesser. So to sum it up, we can say that ajmalicin content increased more than catharanthine. Okay. The next question is, for production of monoterpenoid indole alkaloid, that is MIA, in yeast, three modules were devised which deal with de novo synthesis of intermediates of MIA biosynthesis pathway. Second module mostly deals with the production of dash. Option A windolin, option B catharanthine, option C strictosylin, and option D loganic acid. You can take a couple of minutes. We'll look at how these three modules were incorporated into yeast. So for monoterpenoid indole alkaloid synthesis, this is how the entire pathway looks. First we have the iridioid pathway, and then we have the Taverson to Windolin pathway. Finally we have either Windblastin or Windcustin. Okay. Now in yeast, when this was done, the three parts were streptozidin module expressing iridioid biosynthetic genes, catharanthal or Taverson module that expresses to streptozidin synthase, STD, other ops, finally to generate catharanthal and Taverson. So first we have the iridoid pathway, then that would ultimately give rise to streptozidin. Then we have the catharanthal window, like it was mentioned. Finally we have the Windblastin. So these are the three different modules. So second module mostly deals with option B, catharanthal. That's the major part. Okay. Next question is, identify the specific cell type in Catharanthus roseus leaf where D-acetoxywindolin converts to windolin. You can take a couple of minutes to think about this question. Okay, we are talking about enzyme expression in a specific tissue. Okay, so in Catharanthus roseus, this entire pathway is divided into specific, specific parts of the pathway happens in specific tissues. We have a schematic here. For example, okay, so these genes like PAL, C4H, the entire of the phenylpropanate pathway, multiple of those, it happens in xylem, epidermis. Sorry. What we are looking at in the question is where D-acetoxyvindolin converts to vindolin. Now D-acetoxyvindolin is converted to vindolin using these enzymes, DAT and D4H. This happens in the lattice of fires idioblast. So the correct answer for this question is option C, lattice of fires. Next question is higher molecules of prenyl phosphates are formed by head-to-tail addition of isoprene units. Identify the enzymes leading to the formation of geranyl-geranyl pyrophosphate from isopentenyl pyrophosphate. Option A, isoprene synthase and prenyl transferase. Option B, isomerase and prenyl transferases. Option C, prenyl transferase. Option D, isoprene synthase and isomerases. Okay, so if we look at this, we need to know how these phenyl phosphates are formed and what are the enzymes. So we have dimethyl allyl pyrophosphate and isopentenyl pyrophosphate, which can be interconverted by isomers. So the only difference is where this double bond is. Okay. So if isoprenyl IPP, if that is if gernyl pyrophosphate synthase is present, then we'll get gernyl pyrophosphate. And if farnesyl pyrophosphate synthase is present, we will get farnesyl pyrophosphate. If we have gernyl pyrophosphate synthase, we will have gernyl pyrophosphate. The question is enzyme that leads to the formation of gernyl pyrophosphate from isopentenyl pyrophosphate. So for this, we will need this isomerase. We will also need a prenyl transferase. So in detail we have divided here. So this isopentenyl pyrophosphate isomerase converts IPP into dimethyl allyl pyrophosphate DMAP. And the prenyl transferase catalyzes the addition of isoprene units. So that's what we saw here. One by one the chain is elongated. So for this process we need both the prenyl transferase and the isomerase. Okay. The next question is precursor for terpenoid biosynthesis, isopentenyl diphosphate can be produced via MVA or MEP pathway. Identify the correct combinations of primary metabolite precursor that enters to these pathways to produce isoprene units. And 4 options are given here. Acetyl-CoA, malic acid and pyruvic acid, malic acid, pyruvic acid and shikimic acid, acetyl-CoA, glyceraldehyde 3-phosphate and pyruvic acid, acetyl-CoA, glyceraldehyde 3-phosphate and malic acid. Okay, so earlier we saw about the condensation where isomerase and a phenol transferase is needed. Now they are talking about the precursors also. So they are asking which precursor comes from which pathway. So from the MEP pathway we have pyruvate and glucaraldehyde 3-phosphate. Among these options, option C, which is acetyl-CoA glucaraldehyde 3-phosphate and pyruvic acid, comes from the MEP or the MVA pathway. From the MVA pathway, we have acetyl-CoA. MVA is the mevalonate pathway. From the MEP pathway, we have glucaraldehyde 3-phosphate and pyruvic acid. The next question is match the terpenoid product that derives from its final isoprene precursor unit. Option A, carotenoids. See, column A, carotenoids, monoterpenoids, cis deuterpenes, and D, cytokines. Column B, geranyl diphosphate, farnesyl diphosphate, dimethyl allyl diphosphate, geranyl diphosphate. So these terpenes are classified based on the number of isoprene units they contain. So isoprene is C5 carbon, but it also has unsaturation, a double bond, and also it is not a linear chain, it is branched. Okay. Now carotenoids are C5. They are tetraterpenes. An example would be general diphosphate. Next we have monoterpenes, which is C10, which is general diphosphate. Then we have sesquiterpenes, which is in between both. That is C15, which is farnesyl diphosphate. Then finally we have cytokines, which are the plant hormones. So to this belongs DMA, which is dimethyl allyl diphosphate, which is the initial precursor. It is C5. So based on this, A is 4. It's either option B or C. Then carotenoids, sesquiterpenes is 15, which is farnesyl. Sorry, which is yeah. Sesquiterpene is farnesyl diphosphate, which is C15. So among B and C, C2 is in B. So it's option B. The next question is identify the oxygenated derivative of carotene. Carotenoid from the options given below. Beta carotene, alpha carotene, lycopene, viola xanthan. Okay, so if we look at carotene, we have multiple variations. From lycopene, we get beta carotene. From beta carotene, we get beta cryptoxanthin. We get zeaxanthin, anthraxanthin, and violaxanthin. So here, from zeaxanthin, we have an OH. Carefully look, there is a hydroxyl group here. And in addition to that OH, we also have two of these oxygen linked groups in violaxanthin. So among the options given here, the only oxygenated derivative of carotene is violaxanthin. So in general, carotenoids can be classified into two main groups. The carotenes, which are purely hydrocarbon molecules, and xanthophylls, that is the oxygenated derivatives. This violaxanthin that we looked at belongs to xanthophylls. Next question is, identify the correct step in the carotenoid biosynthesis that is not catalyzed by lycopene cyclases. Option A, trans lycopene to gamma carotene. Option B, gamma carotene to alpha carotene. Option C, beta carotene to beta cryptoxanthin. Option D, gamma carotene to beta carotene. So lycopene cyclases perform multiple steps. So they are of multiple types as well. So the lycopene epsilon cyclase forms delta carotene, whereas lycopene beta cyclase forms gamma carotene. Then lycopene beta cyclase can form alpha carotene. Lycopene beta cyclase can form beta carotene. Now the question here is the correct step carotenoid biosynthesis that is not catalyzed by lycopene cyclases. Here, one of the other carotenes are being formed by lycopene cyclase. Only beta cryptoxanthin is not being formed. Beta cryptoxanthin is formed by beta carotene hydroxylase. So, that is the step. So, the explanation is given here. These are enzymes that convert lycopene into cyclic carotenoids like gamma carotene, alpha carotene, and beta carotene. And they convert trans lycopene or lycopene into gamma carotene, gamma to beta, and gamma to alpha. Right. The next question is match the anthocyanidin pigments in the left side with their correct coloration given in the right hand side. Color coordinated cyanidin, petinidin, and malpidin. And the colors are red purple, reddish purple, and orange red. So here also it follows a spectrum. This is a gradient. So this is how the coloration looks like. At the left end of the spectrum, which is towards the orange reddish, you have color guanidine. On the extreme right, you have malguanidine. Malguanidine is blue. The next question is an application question. You want to create blue roses. What are the major requirements to achieve this goal? 1. A relatively low vacuolar pH less than 4. 2. Synthesis of delphinidin. 3. Presence of flavonol copigments. 4. Synthesis of cyanidin. 5. Relatively high vacuolar pH. So to be able to answer this question, we need to first know which of these colors, pigments, delphinidin or cyanidin is blue or close to blue, and at what pH this could be stable. Think about this question in terms of these two aspects. We'll try answering the question. So the first thing that we need to know is which of these colors correspond to a bluish tint, whether it's cyanidin or delphinidin. Cyanidin is pinkish, delphinidin is blue. The next question is which pH would favor this. So higher pH, acidic pH would favor red, higher pH would favor blue. So now we know that 5 and 2 are present in the right answer. And in addition to stabilize these pigments, we also need the co-pigments. So option C, which is delphinidin, flavonol co-pigment and high vascular pH is essential. Next question is, identify the group of compounds in which resveratrol belongs. Stilbene, charcoal, anthocyanone and aroma. So this is the general structure of flavonoid, where we have a chalcone has two rings that are interconnected here, whereas flavone has these the phenyl rings that are interconnected, and this is also cyclic. So the biphenyl. You have dihydrophenol where you have these overheads, then leucoanthocyanidin is a part of this. Now, stilbene is this where you have this two rings connected by a C2. So it has this general skeletal structure of C6, C2, C6. Oron is close to him. So we are talking about resveratrol, right? This is the structure of resveratrol. So it belongs to stilbene, where you have C6, C2, C6. You also have additional OH groups, and is classified as a phytoalexin. These are produced by some plants in response to stress or pathogen attack. We saw this question earlier. Next question is, select the bacterial species that are capable of utilizing ferulic acid as sole carbon source. 4 options are given here. Species of Pseudomonas fluorescens, species of Pseudomonas aeruginosa. So if we look at this, in this paper published in the early 2000s, we observed that when this mixture is incubated with cells and then ATP, magnesium, OSH, what happens is the ferulic acid levels diminish to produce vanillin, vanillic acid, and the presence of NAB, which is the cofactor that is needed that gets reduced and the product gets oxidized. More of vanillin gets converted to vanillic acid. So ferulic acid getting converted to vanillin and vanillic acid, and this happens in Pseudomonas fluorescens AN103. The next question is about Shikonin biosynthesis. We have to identify the fall state. So they ask about the color of the compound and the side chain, the pathway, and whether it has a pentenyl ring or a prenyl ring, and so on. So Shikonin is just a red color pigment. So it has precursors from both the phenylprotonate pathway and the mevalonate pathway. So we have 4-HPA here and GBP here. Now, the Shikonin has a prenyl group, which is a 5-carbon compound. Okay. It's a prenylation step, and it is not a pentenyl side chain. So the false statement here would be a pentenyl side chain is attached to the aromatic ring. The next question is, scientists have successfully transformed HCHL gene to produce vanillin in non-vanillin producing plant. Upon time course HPLC analysis of the plant extract, a gradual decrease was observed in feryl oil CoA concentration. Identify the correct statement from the following list based on the given information. Option A. The expressed enzyme is capable of utilizing feryl CoA acid substrate. Option B. The expressed enzyme is not capable of utilizing feryl CoA acid substrate but can produce vanillin. Option C. The expressed enzyme is capable of utilizing the cinnamoyl CoA acid substrate. And option D. The expressed enzyme is capable of utilizing both feryl CoA and cinnamoyl CoA acid substrate. So here we are talking about HCHLG. It is the hydroxycinnamoyl-CoA hydratase lyase. It would convert ferulyl-CoA into vanillin and acetyl-CoA. So they say that upon time course HPLC analysis, there is a decrease that is observed in ferulyl-CoA concentration. That is because it's a substrate. That is straightforward. The last question. Chiconin is a red dye naturally produced in flowering twigs and root of Lithospermum erythrorhizum. Identify the most suitable choice for industrial production of this red pigment from the following list. Option A, shoot culture. Option B, hairy root culture. Option C, adventitious root culture. Option D, cell suspension culture. So like we discussed this yesterday, Chiconin dye when conducted through the cell suspension culture produces this red color pigmentation. So these cell culture are grown in two stages. First is the growth stage where Mg5 medium is required. Next is the Chiconin induction medium, which is nothing but the Mg9 medium. Here the cells are grown and then they are allowed to produce Chiconin. So with this we come to an end of the extra session. Thank you for attending.